Second.
All right, well, while we're starting it up there, my name is Kieran Normoyle. I'm from Mass General Hospital. Thank you, Sarah, and everybody, the organizers here, for inviting me to speak. Thank you all for your interest. So I'm going to talk a little bit about the basic science of GABA-A receptor channels. Dr. Rogowski made this pretty easy for me by already giving a pretty good intro to that. So I might just kind of go through that a little bit. My area is actually on my area of study is on more chloride homeostasis. So I'll kind of spend a little bit more time on that, I think. So the structure and the function of the GABAergic system, the primary system of inhibition in the human brain, is really the focus of what we're all talking about today. And it's already been pointed out that the GABA-A receptor is a pentameric receptor, typically of 2 alpha, 2 beta, and a gamma. You can see here how there are actually 2 GABA sites. As well as another separate allosteric site that benzodiazepines are typically bind at. I think I'm kind of preaching to the choir here, so I'll kind of move on from that. What the really interesting thing about this, when we really think about the way in which the GABA receptors or these types of receptor channels are so intricate and interesting. Is the fact that they have to do several things at once, right? These binding sites that you see, where GABA and diazepam actually bind, are very external, and they're but yet they're dictating through longer distance conformational changes. What happens at this, for instance, right at the pore, where the actual selectivity for chloride is happening, and what that ends up with is that you can't, at first glance, as a biochemist, you might think, well, if we have a mutation that's in one general area, that should tell us if we're in the transmembrane area versus the N-terminal, extracellular area of a GABA subunit. That should dictate the phenotype. And this really breaks down for the fact that there are long-term conformational changes that do occur. It becomes something that's very difficult to really kind of nailed down in that way. I'm not going to walk you through this. I think that we're already quite, if there are any questions about that, just let me know. So here I'm just kind of showing the generalized, expression and trafficking. Each one of these are potential points at which we could, think about treatment. Obviously, there's your transcript is actually in the endoplasmic reticulum, getting inserted into the membrane where it should properly fold. Then in the case of GABA, 5 separate subunits will then assemble into a given pentamer before being transported to the extracellular membrane. Those are all places where things can go awry. But in the case of GABA, what tends to happen is if you have a mutation that affects the suitable, the suitability of a given receptor, you can often have simply a different typical conformation, or the makeup of a pentameric GABA receptor can actually change, and it gets even more complicated. With Dr. Kang's work. Has shown that even if the mutation is in, say, the gamma-2 subunit, which particular beta subtype is present, can affect the trafficking of that pentamer. So it's very complicated, and I am maybe during the discussion, if anyone has questions about that particular thing, I think that there are other people in the room that are probably better able to talk about those specifics. But suffice it to say that this ends up being something where we have a large spectrum. This again from the work of Dr. Kang and McDonald, where you can have a full range of phenotypes from simple febrile seizure to Dravet syndrome, just based on where that particular mutation is occurring. So the reason for I'm going through all this without really delving into the specifics of one mutation versus another, because they all feed into the same basic physiology here. Is that GABA at the end of the day is conducting chloride across the membrane, and very importantly, is this is intertwined with and connected to intrinsically the function of the GABA receptor, that is, of GABA neurotransmission. And it is that that difference between the extracellular and intracellular chloride concentration that's really the key here. And that is about a tenfold difference, by the way. Really what it gets down to is, you'll be familiar with the Nernst equation. Of course, it's a little bit more complicated in the actual neuron, because there are several different ions. I'm only looking at chloride. And there's also an osmotic potential. I'll talk in a minute about the Donin potential. But it's what it really comes down to is what is the concentration of chloride inside and outside the cell. And for a number of years, we felt that is, we as a field, that we know the answer to this. For intracellular chloride, for good reason, there's been a lot of work into looking into how to measure that, and there are good tools to do that. Where there are not good tools currently is extracellular chloride. It's actually very difficult to measure that without causing damage. To do that non-invasively, to measure extracellular chloride, and so that's what I'm kind of focus on a little bit here. I just want to point out one last thing, is that this the assembly, folding, and transit is actually important for other types of transmembrane channels and receptors, most notably cation chloride cotransporters, which we're going to talk about a little bit next here. So the eGABA, that is the reversal potential of GABA, the potential at which the direction of flow of chloride across GABA actually changes, is not set by the resting membrane potential of a neuron, although it is near the resting membrane potential. I think that's a very important concept that has at times taken for granted. The prevailing theory of what does set eGABA has been since around for about 20 years, since Richard Miles' studies, which showed in resected human subiculum that in epileptic tissue you actually had a high intracellular chloride, and there wasn't KC2 being expressed. The idea here was that as cells mature, they go from a neonatal common state where KC2 is not expressed, and therefore the idea is, well, you have a high chloride as a result, to a more mature state where KC2 is expressed, and now you have chloride extrusion, and this is how you get a mature cell. Neuron rather, and what that causes an actual change in the eGABA, such that you go from an immature depolarizing GABA and activating GABA response to the more canonical hyperpolarization. However, this kind of is a very nice way of looking at it, but it turned out that when Miles looked into this a little bit more, the data didn't actually support this. So whether or not KC2 is actually present doesn't dictate whether or not you have a depolarizing or hyperpolarizing GABA response. Furthermore, in trying to figure out this conundrum, when you actually look at let's try to characterize then how important each of the cation chloride cotransporters and KCC1 and KCC2 are in setting that membrane potential, in the GABA reversal potential, and you actually take a look at using chlamylion here, which is a ratiometric intracellular chloride reporter. What you find is that the best when you block one or the other in sequence, the best predictor of what the final concentration will be is what the initial concentration was. And the surprising thing about this is it doesn't matter if you're looking at neonatal neurons or adult neurons. It doesn't even matter what which one you block first or second, and it doesn't matter what fluorophore you're using. This data here is using a separate fluorophore altogether, clofensor, that's measuring intracellular chloride, and you see that the trend is the same. I should note that these authors interpreted their data a little bit differently, but clearly these are not what's actually setting eGABA. So if not rapid equilibrative CCCs, then what is setting eGABA? And the important thing that I want to note here is that we found that there's actually intracellular microdomains that we find inside of neurons. This work was published in 2021. I'll come back to it in a little bit. But what I really kind of want to talk about, the theme of compensatory changes has come up. And I'm very glad that it has, because that's really kind of what we're focusing on here is, that the reversal potential of eGABA and how neurons are actually, they're in control of how the GABAergic signal is being interpreted by the neuron. And so I'll explain a little bit about what I mean by that. So I told you I was gonna get back to the donut equilibrium, right? So there's fixed anions inside and outside of the cellular membrane, and this is really important because on the extracellular side, you have principally chondroitin sulfate. They're glycosaminoglycans that have an immobile anionic charge that forms the immobile phase of a donut equilibrium, and inside the cell there are several examples of candidate molecules for an immobile donut effector. And I'm going to focus on actin in particular, which when polymerized actually has a linear charge, or a charge of about 4 negative per linear micron, which adds up when you have a large biopolymer. Because it's also a highly cross-linked branched network, it's relatively fixed in position. And so for our purposes, it forms the immobile phase of a donut equilibrium, which, like a photonegative, will in turn dictate what the concentration of chloride actually is. So the neurons are not directly telling you exactly where the chloride can be or what it is, because it's going to diffuse. But by dictating what where the immobile anions are, it's able to do that. And so this is how the neuron is able to have these intracellular microdomains. So I've shown you those. But then we want to, I will show you those. But we wanted to know about the extracellular chloride as well. Is it really 100 millimolar, or roughly 100 millimolar? The concentration of chloride is on the outside of the cell, given the ACSF in our experiments, or the CSF in patients. It turns out that it's not. And we shouldn't expect it to be, right? It's surprising that it's not, but we probably shouldn't expect it to be. Why is that? So the donut effect is, I'm going to show you some experiments briefly where we are taking a look at an artificial extracellular matrix. So what we did here was we used agarose. So many of us have used agarose in the lab, right? It's it's going to form a gel that has the important thing for if you're trying to separate anions like DNA is that it doesn't actually have any anions of its own. And so what we did was we chemically put them there. So we made a sulfated agarose that ended up having about 50 millimolar worth of covalently added sulfate. And then what we did was we demonstrated that that chloride is actually excluded from these sulfated gels. So you can see that the concentration of chloride is going to be inversely proportional to the concentration of sulfate in the gels. Here's the ACSF bath, regular agarose gel. There's no difference. And then whether we're measuring with a chemical colorimetric assay or with our I haven't actually introduced it yet, but ABP dextran. our new fluorescent indicator, we find that there is significantly less chloride within the gel. And this happens if we do an experiment like this in the schematic where the gels are just sat next to each other. You see the same thing. And we can leave that for up to more than 3 weeks and it's still the same. This is because the sulfate is covalently bound, the same as chondroitin sulfate is covalently bound in the extracellular space. And so what I really want you to take away from this, benchtop, ECM experiment is that it should not be surprising that the concentration of chloride in the extracellular space is significantly less than it is than is the bath that's there, the CSF or ACSF that you've provided. And so I'll show you that we've actually measured the concentration of chloride inside and out. I'll just briefly show you this in the inside of the cell. And I want to show you here that it's actually actin that's causing this. So our control versus a chloride insensitive fluorophore, there's no difference. Whereas if we disrupt actin with latrunculin B, which importantly causes a disruption of actin that does not cause disruption to the structure of the cell, we see that there's no longer a correlation between the pre and post chloride microdomains. But importantly, this is just showing here that these are those ones are not significant, but that the change, the overall change in chloride is nothing. It does not change the overall chloride concentration when averaged across the cell. So what's happening there is that because in these experiments we've actually blocked the cation chloride cotransporters, and we've blocked seizure activity with TTX. Chloride isn't moving back and forth, but it is homogenizing. So I hope that this proves to you that we are actually seeing these microdomains and that we have control of them using latrunculin B. Neurons have control of them under regular normal physiological conditions. Joseph Glikas and Kiyoshi Igawa, former lab members of the Staley group in Boston, had already shown through alcium blue staining. This is very invasive. It requires fixed tissue. But they showed that there was an anticorrelation between the amount of negative charge that's being shown here by alcium blue, the amount of extracellular sulfate, basically. As that goes up, the intracellular chloride goes down. The idea there was that in this hippocampal slice model, that what we're seeing is that where there's more sulfated glycosaminoglycans, you're going to have less extracellular chloride. But we needed to be able to measure that. And so the way that we were able to go about doing that was with ABP dextran. I'm happy to answer questions about it, but I'll just say that we used fluorescent lifetime imaging, which allows us to have a concentration independent way of measuring the chloride using this particular dye. And what you'll see here, I'm just going to show you briefly that in hippocampal slices, you can see that the ABP here, you're actually looking at the concentration of chloride is less. I'll show you here with the histogram that the median here is actually 44.5 millimolar. The ACSF surrounding this slice is 136. Right, so that's nearly a deficit of 90 or so millimolar chloride. That's a lot of chloride that's been displaced, right? And that's going to have a profound effect on how chloride is handled and how chloride is transmitted at the time when GABA is activated rather. And here I'm just showing you a perineural trace. You can see that there's an awful lot of difference in change in chloride per micron. There's a lot of variability as you go around the outside of the neuron. Just a functional test of this is that, what would happen if you, like my electrophysiologist colleague, Nega, patched a pyramidal cell and then puffed glutamate on a bunch of interneurons that were synaptically connected at different spots of that pyramidal cell. And the answer to that, she found, is that you get a bunch of different GABAs. And this is because they're all connecting at different spots. There's presumably a different intracellular chloride and different extracellular chloride at each one. I won't go into that too much. And then I just want to show you that we've demonstrated that this is happening intracellularly. Here you can see that ABP does not go into vasculature nor into cells, but when we measure the extracellular, here just showing the RIs, and when we measure the extracellular chloride, you can see here the control, it's 136. Whether the fields are cellular or acellular, when we combine all fields, we combine all animals, we still end up with roughly 50 millimolar chloride in the extracellular space. We've also shown this in, this actually is data coming from a piglet model. So a gyrencephalic model, as opposed to our lissencephalic mice. And you see again that the control animals have an extracellular chloride of just under 50 in this particular animal. And then after injury, when MMPs are being activated, and we our hypothesis is that it's the remodeling of the extracellular space and the breakdown of the glycosaminoglycans, that you then see that chloride is able to increase. And we show that the intracellular chloride goes up as well. And so I want to talk about the potential impact on GABA-AR function, and we'll leave this for the discussion. I just thrown this guy up here. Just imagine now if we, I just put up several of these here, that individual, other factors that as we talk about how much GABA is released in the synaptic space, as we talk about what's going on in the postsynaptic side and in the presynaptic side, if there's a fundamental change to what is the extracellular intracellular chloride, that changes the system quite a bit. And the neuron has the means, every neuron has the means to compensate for those changes that are happening because of a genetic incident. And just Dr. Rogowski actually had this yesterday, this lovely slide. And I would just add to the idea that these are anti-epileptic, that we need anti-epileptic drugs. I just point out that the refractory patients, when we had our first generation drugs, were 30%, but that's fine because we had second and third generation where it remains 30%. And I would just point out for our discussion that the fact that we still have 30% refractory patients is rather an indictment of how we're finding targets, and the way in which we are looking at targets. It means that we're missing something. And so I hope that maybe you'll take from this that maybe understanding exactly what the concentration is locally at a GABA synapse is something that we maybe should know a bit more about, or spend a little more time thinking about. So I'm happy for the discussion in that. And I'll just put this slide up here to mention briefly, as Kevin Staley is our leader out in Boston, and you can see all of my wonderful colleagues here, some that are not pictured, that all had at least something to do with this work. I'll point out Kyle here, who does the piglet model. It was his data that I was showing. So thank you very much, and I look forward to our discussion.